So this student has completed the mitosis lab and answered the questions that were posed to them in that lab. And remember, the lab says after 5 cycles, predict how many chromosomes you're going to have inside the cell. And I sent you a video that was intentionally vague, and so that you would think your way through the problem and come up with the answer yourself. And as you see here, the student has done just that. So these are the number. This is a cycle number, and Samantha, and I'm not putting last names so that because it is a video, I'm putting it on YouTube. Samantha put down her cycles 1 through 5, and every time you went through the cycle, you go through all 4 steps of mitosis. And remember, the problem is what would happen if you used mitosis for reproduction rather than meiosis. And so what would happen is that you would divide, and remember in mitosis, you divide the cell into 2 cells with equal number of chromosomes. And and when you combine those 2 cells, one of those 2 cells with someone else's cell that went through the same process, it's going to be different than what would happen during meiosis, because in this situation you're using mitosis. So here is Samantha's workout. Here's a cell with 2 chromosomes. It starts with 2 chromosomes, just like the problem says, and it goes through DNA replication. It has 2 chromosomes. It started with 2, ends with 2. That's mitosis, right? 1 from mom, 1 from dad. Ends up with 1 from mom, 1 from dad in both cells. So now, and this cell also did it. So this is one parent. This is another parent doing this, creating their gametes in this wrong way, right? This is not the way it goes. So this problem was about what would happen if instead of meiosis, we used mitosis to create gametes and then to create these new beings. So these 2 cells then are fused, and it creates this cell. Because you didn't have the DNA, right? Because you didn't go from 2N to 1N, each of these still has 2N, so you end up with 1, 2, 3, 4, right? 2 blues and 2 pinks, 4. So that's where you end up after cycle 1. After you go through the cycle again, of course, it does mitosis again. Mitosis means the exact number of their exact copies, right? So if you started with 4, you're going to end with 4 in each of these. And so you go through the 4 phases of mitosis, and you end up with 4. The same thing was happening over here with on this side with these individuals, with these 2. So this cell then with 4 chromosomes and this cell with 4 chromosomes fuse, and now you have 8. And you see that in her model, this is her model. She drew it out, didn't she? Now, you could have used popsicle sticks. You could have used whatever you want. You could have videotaped it. However you wanted to handle developing your model. But in her model, she drew out these cells, and each little line is a chromosome. So now they're fused, and now instead of 4 chromosomes in each cell, now you have 8. Now you go through mitosis again, and each cell has 8. And they don't really fit, as you can see here. She's saying, the second fusion. So here's your second fusion, right? And you're getting now you have, instead of 8, you do mitosis again. You end up with 8 chromosomes. But now these 2 are going to fuse, and now you have what? 16. Again, going through mitosis, dividing those 16. There's 16 chromosomes in each cell. You fuse those 2, you get 32. And again, you do mitosis. So now these 2 cells have 32 each. You fuse those 2, you get 64. So that's what would happen after 5 cycles. This is your data. After 5 cycles, you go from 4 to 8 to 16, 32, and 64. Now, I asked you, if you wanted extra credit, you could take this data and graph it and extend the line and predict what would happen after a certain number of cycles. So here's her graph that got her the extra credit, and it did a great job. She did a great job doing it. There's a few minor mistakes, but one thing I'm kind of always hesitant to give full credit for any graph that doesn't have a title, an axis title, and so it would have been nice to see a title here on the y-axis. So we know that this is a fusion number or cycle, right? And it doubles every fusion. So every time it fuses, it doubles. So you should see, very good. This is actually the model. What you're looking here, this is the model. This is a theory. So this is quite valid. And here you see it doubles every time. And she estimates that at 8 it'll be 256. At 9 it'll be 512, and at 10 it would be 1024. At 6 it's actually 128, and then 7 would be somewhere around here, and that would be double 128. So double 128 is 256. So you'd probably want to move that over, and so the numbers here are a little off, but not a big deal. I think she did a fantastic job overall, and this is exactly what I was looking for. I hope it wasn't too difficult for you. I'm going to wait to send this tomorrow as an explanation to the lab. I expect that everybody is giving it a shot, and you come close to something like this. As you saw, the real intent was to look at why is it that we need meiosis. There's two reasons. We've discussed them in class. You've read about them in the chapter. You've read about it in the project that you have to complete by Thursday, and we've gone over it in class. There's two reasons. You have a 2n number, where n is the number of chromosomes in a diploid organism. Diploid meaning an organism that has two copies of every chromosome. As I said before, not every organism has that, but in humans we are 2n, we are diploid. And the whole point of meiosis is to take that 2n and bring it down to 1n. So take 2n and bring it down to 1n, so that when you do that and you meet your spouse, and you want to have a child, they can take their 1n cells, which we call what? I hope you said gametes, right? Which would mean sperm or eggs in humans, right? You take this sperm and this egg, and you fuse them together into a new human being, or new human being. An n plus an n, two n's come together to make a 2n. So you started with 2n, and you ended with 2n once you combine these two different set of genes. Not doing this, not reducing the chromosome number, as you can see, quickly gets out of hand. So only in six matings you come up with 128 chromosomes in this cell, rather than just whatever the chromosome number for that species is. And also would be 46 or 23 pairs or 46 chromosomes. But here, instead of 46, you'd have 128, only after having only 6 different fusions. This is a fusion, right? A sperm and an egg. Only having 6 different fusion events, 6 different combinations of sperms and eggs. Would you get then to 128? And if you went 7, 8, 9, 10, you'd quickly come up to thousands of chromosomes in a cell. And that's just not that's not a workable model. That's not a workable way of doing things. So this model, where you're taking 2n and breaking it down into 1n each by doing 2 cell divisions, right? This model is the model that we actually use and that works. The other reason for meiosis, as we said in class, and I'll post that video before you'll see this one, and the other thing that is an advantage in meiosis, and in fact is when we say an advantage of sex, right? When we talk about sex, we're talking about recombining the DNA and combining them, male and female, so egg and sperm coming together. What's the advantage of that? Is that you're increasing genetic variation. What we don't want is to have a small gene pool. The bigger the gene pool, the bigger the variation in a population, the more likely it is to survive any catastrophic event. So the idea of increasing genetic variation is a really big advantage of sexually dividing creatures. Bacteria and other asexually dividing creatures have a limited set of genes, and even though bacteria have a way of recombining their genes uh, through something called conjugation, which is similar to sex in the sense that DNA is shared between two organisms, genetic variation occurs in meiosis in two ways. Number one, during actual conception, when the two gametes come together to form a new organism, when the one end gametes come together to form a 2N, you've combined genes from two different organisms. So that's one way we increase genetic variation. The other way we increase genetic variation is through that process of crossing over. And I'm not going to go into it because we went into it in class, and it'll be in the video as well. So I don't want to get into it too much. But genetic variation, stems not just from the idea that you get genes from two individuals coming together to make a new person. You also have this idea of crossing over, which then recombines that DNA. All right, so this model was perfect, as I said. As far as I'm concerned, conceptually, she got the point, which is that if you don't use meiosis, this process of fusing two gamete cells becomes unmanageable. And her answers are here. As you can see in the answers to the questions that she had written, she chose to submit her work on her lab via pictures, JPEGs from her phone, and then she wrote in the Juno doc. She actually wrote the answers to the questions. As you see here, when it says turned in, this is what I see when I grade your work that you submitted. This is the work. The JPEGs that she submitted are here. And these are the answers to the questions that she submitted through Juno Docs. You can see that because it's in Juno Docs, I can highlight and make comments directly on the document, and you can see that I can quickly grade this. I know how many words, how many characters, etc., and when it was last modified. So. Yeah, this is the correct answer, right? The chromosomal number from each previous fusion is being multiplied by 2, so it's doubling every time there's a new fusion. And that's how chromosome numbers in any model is constantly changing with every cycle of mitosis and fusion, right? So that's answering the question. In order for the chromosome number to stay at a constant number, you need two cells to be fused that have the same chromosome count. Very well said. So you need N and N to come together and form N. Right? So I believe I understand what she's saying based on her work that she submitted. This answer here is a little bit vague, but if I combine it with the work that she submitted already that we just went through, then I think I understand what she means. You cannot allow the chromosome number to continue to change from one generation to the next. They have to stay the same. Otherwise, you're going to get uncontrolled increase in chromosome number. So I believe that's what she meant to say by the same chromosome count. Bottom line, in order to get that same chromosome count, generation after generation after generation, you have to half the DNA. You have to go from 2N to 1N. Why do we use 2N instead of just saying go from 46 to 23 chromosomes, 46 pairs to 23 single copies? Why do we say that? We say that because different organisms have different numbers of chromosomes. But diploid organisms always have two copies of each of those chromosomes, whatever those numbers are. One from their mother, one from their father. So in humans, it's 23 pairs. Maybe in X species or X tree or plant or animal or insect or animal or what have you, instead of 23 pairs, maybe it's 2 pairs. Maybe they only have 2 chromosomes. Or maybe they have 100 pairs. But whatever it is, this number, this total number of chromosomes it has in the autosomal cells, are called the 2N. So when these creatures, the humans, make gametes, make sperm and egg, each sperm, each egg has 23 single copies of chromosomes, right? 23 singles. Instead of 46, went from 46, which is the 2N, to 23. 46 pairs of 23 singles. In this species over here, you'd go from 2 pairs, 2 chromosomes, but one from mom, one from dad, to 2 single chromosomes. So you went from 4, right, to 2. Because this is 2 pairs, and 2 pairs mean 4, right? So here's 100 pairs. So you went from 100 pairs to 100 single. So you went from 100 pairs, 200, to 100. In each case, you went from 2N to 1N. Right? You're going from 2N to 1N. So that's why we use this variable. It's important to understand that when we're studying biology, humans are not the only ones that we're concerned with. There's many different species and many different chromosome numbers, and even many different ways of doing this. There are certain models with corn, for instance, where the number of chromosomes does go up and then it shoots back down again, and it's all very complicated. But in the most simple form, in general, diploid organisms go from 2N to 1N in order to make the gametes, so that they can fuse them, so that they can make an organism with 2N. The whole objective is after fusion that you have the same chromosome count from one generation to the next. So well done, Samantha. Excellent job. I hope everyone follows suit and does a similar work. All right, bye-bye.